Let me just introduce myself first. I'm Jordan Smoller. I'm based at Mass General and also part of the Stanley Center for Psychiatric Research here at the Broad. And I have the pleasure this morning of talking with you a little bit about the issue of phenotype definition. And for those of you who have been coming to the primer over the last several weeks, you know that you've seen an array of phenomenal speakers talk about the advances in genomic medicine that have happened in the last 10 years or so. And it really has been pretty spectacular. We can now do sequencing shortly at a rapid, high throughput level. In some ways, we haven't, I think, spent enough time as a field, but also this is probably a little bit more than you've had in this primer about the other side of the equation, that is the phenotype side. So anytime you're doing a study, a genetic study, whether it's a linkage study or an association study or a sequencing study, you're essentially trying to connect some genotypic variation to a phenotype of interest. And in some ways, the dramatic advances that we've had on the genetic side have outpaced some of the thinking about what's the best way to think about phenotypes to maximize the power of our studies, the interpretability of our studies, and so on. And that may become the rate-limiting step for some of the advances that we have or that we're awaiting in the future. And that's a little bit what I want to focus on today and talk about some issues about phenotype definition with tales from psychiatry, as I say here, in part because, and I like to say psychiatry is sort of the New York of phenotype definition. If you can make it there, you can make it anywhere. It's incredibly complex. As we'll talk about in a couple of examples, the phenotypes are arrived at this point largely by the consensus of experts. So as opposed to diabetes or autoimmune disease and so on, where we can actually look at biological markers or pathology, we don't have that advantage in psychiatry. And so phenotype definition becomes part of the art of moving forward with genetics. And let me just see if I can, oh yeah, here we go. Okay, so let me just talk a little bit about some of the issues that I want to cover in the next 45 minutes or so. And I will encourage you to ask questions as we go along. So the first issue is how we select a phenotype. How do we begin to decide that something is worth studying as a phenotype? And we'll talk about some of the issues there. Secondly, once we've decided on a phenotype, what do we do about measuring it? How do we decide what's the best way to quantify or categorize the phenotype of interest? A couple of things about refining phenotype definition, the idea of using subtypes or what sometimes people call intermediate phenotypes that we will talk about as well. And then the last thing I want to just mention is in some ways turning it around, and that is using genetic information to inform our understanding of the phenotype. And as we'll see, that can get us into a kind of iterative cycle, which may be very interesting and increasingly relevant in the years ahead. So that's where I want to focus us today. So let's start out with this idea of phenotype selection. Now in psychiatry, in some ways, we're extremely lucky. If we want to know how to define our phenotypes, all we do is we go to the bookshelf and pull this book off. This is the DSM-IV manual. And basically, this is a catalog of the diagnostic criteria for every officially recognized psychiatric disorder, at least according to United States psychiatry. So we just need to look it up, and that's it. That should be pretty easy, right? The problem is that exactly where to draw the line around a phenotype is incredibly complex and somewhat a moving target in psychiatry. And you may just know this from your experience of hearing about psychiatry, but this is a picture of what this phenotype manual has looked like over the years. So here's the DSM versions. It first came out in 1952, 1968, 1980, 87, and 94, which is the latest edition. And what you're looking at here is the number of pages in the book and the number of diagnostic labels. So you can see we've been enormously successful in identifying new phenotypes, if not the genes underlying those phenotypes. So people worry that maybe almost anything could be a psychiatric or behavioral phenotype, and that undermines our confidence to some degree in what we need to focus on. And you can get into this kind of situation. This is my favorite example always of these kinds of phenotypes. When you're on the phone with somebody and they say, I'll let you go now, like they're doing you some kind of favor, but it's really them that wants to get off the phone and they just don't want to admit it. I think I've isolated the gene for that. A lot of people think that's, in psychiatry, what we're aiming for, and of course it's not, or at least it's several years away. So the question is, what is really a reasonable phenotype? And how do you decide that? Let me give you an example of the kind of thing we're talking about, the kind of decision you have to make when you're reading the literature. So here's a study about gene polymorphisms associated with creative dance performance. Is that a reasonable phenotype? Maybe. There is actually a biological model involved in understanding how these variants might lead to the dance phenotype. Well, how would you decide, actually, whether that was a reasonable phenotype? I would say that there are at least a few questions that you want to start asking. First of all, is there any reason to believe that some version of this phenotype is influenced by genes? And if so, a refinement of that is, what's the best phenotype to choose? What's the best measure of that phenotypic trait in terms of its likelihood of being influenced by genes, which you're trying to find? How complex is the genetic architecture? That turns out to be actually the most fundamental question if you're trying to decide what's the likelihood that you're going to be successful in identifying variants influencing whatever this trait is. So those are the questions I want to run through. So let's start with that first question. Is some version of your phenotype influenced by genes? How would you decide that? What's the first question you might ask? Is it heritable? And even before that, or an early view of that is, does it run in families? That's often the first thing, because is it heritable sometimes requires a slightly more complicated study design. So people often start with family studies, and they're asking a simple question. This is actually just a case control study. You take people with the trait, let's say it's a diagnosis of depression, let's say, and you look at people with the trait and people without the trait, and you compare the frequency or risk of that trait in their first-degree relatives. If it's significantly higher, if the ratio of that prevalence is significantly greater than 1, that suggests that it is aggregating in families. And we often talk about the recurrence risk ratio, or lambda. The sub R means whatever relative type you're talking about. So let's say lambda sub S would be the sibling recurrence risk ratio, and that's the risk to relatives who are siblings in this case, compared to the population risk. How much more likely are you to be affected if you're the first-degree relative or the sibling of somebody with the trait compared to somebody pulled at random from the population? And that is an index of the familiality of the trait. So if you look at what some of those numbers are for traits you're probably familiar with, and I've loaded this up with psychiatry, but take a Mendelian disorder like Huntington disease, it's not in some ways all that relevant there, because we know that's a single major gene, highly penetrant dominant mutation, cystic fibrosis. But then you get to things like autism and other complex traits, and the numbers are lower, but certainly significantly greater than 1, suggesting that these things do aggregate in families. But what's the problem with that? Is that enough to say that genes are going to be involved? No, because things run in families that are not necessarily heritable or influenced by genetic variation. Although if you actually think about it, I say that, and then I always try to give an example of that. It's not that easy to come up with an example of that, or at least one that you plausibly couldn't have a genetic component to. I mean, you think about infectious diseases. A cold can run in families. Even there, susceptibility might have some genetic component, but let's assume there's a paper that was just released today about genetic influences on liberal political ideology. So people are extending this, or at least the plausibility of that idea, quite a bit. But it's important to recognize that the lambda, if you're going to use that as your index of how familial things are, can be influenced by things that would bias a case control study. Ascertainment bias, over-reporting. Obviously, if people who are in an affected family tend to report their affection status more frequently than people in the population, that could inflate it. But of course, there's also this notion that it is a ratio, as I just showed you, and so it's going to be sort of bounded by the population prevalence. If the population prevalence is high, then lambda can't be too high, because you're asking what's the relative risk, in a sense, and you can only have a prevalence in relatives of 100% maximum. So Alzheimer's disease is an example. The lambda S is about 3, and that doesn't necessarily tell you that it's not as tractable a genetic disorder as, say, autism, where the lambda S is much higher, because first of all, Alzheimer's disease is very common, so relatives have an increased risk, but they can't have a 100-fold increased risk. But the major issue that I think that is obvious to you probably is that, as we talked about, lambda is a function both of genes and environmental influences. So it's not really telling you how much of the genetic variation you're going to find. So how do you then go on to ask this question about how heritable it is, that is, how influential are genes in determining risk differences among people? And there you want to look at things like twin and adoption studies. You can do these studies in extended pedigrees and estimate heritability, but that's less commonly done. So take the twin study, which is a workhorse of this, at least in psychiatry, but it's also been done in many medical disorders and other traits. And of course, what you're doing here is making use of the fact that there are two types of twins: monozygotic or genetically identical twins and dizygotic twins who are essentially like other siblings. They share on average half their alleles. And what you're doing here is saying, let's hold the environment constant and vary genetic relationships and see how similar are these pairs of people. And the notion is that if the concordance rate or similarity between the genetically identical clone people is greater than the similarity between people who are just, on average, 50% genetically similar, that implies that that greater phenotypic difference can be related to their greater genetic similarity, and we conclude there is heritability here. As I implied, you have to assume something. That is that you're holding the environment constant, right? So if it turned out, as some people say is actually the case, that identical twins are treated more similarly to each other than non-identical twins, they're dressed the same, they're talked to the same, or whatever, that could inflate the apparent heritability because it could create influences that make them sort of spuriously more phenotypically similar. Of course, if the identical twin concordance rate is less than 100%, that sort of immediately implies that genetic similarity is not explaining everything. And then we get this index, this metric, the heritability, which is often misunderstood, but which refers to the proportion of phenotypic variation in a population that's attributable to really genetic variation. You can sort of think of it as how much of the prevalence, that is the difference between being affected or unaffected in a population, is attributable to genetic differences among people. And that is a number that can be expressed in a couple of ways. People talk about the narrow sense heritability, the variation due to additive genetic effects, or the broad sense, which would theoretically include both additive dominance and gene-gene interaction or epistatic effects. It doesn't really account for gene-environment correlation. That is, and we see this actually in behavior and psychiatry. People, because of their genetic background, may put themselves in environments or expose themselves to environments that are influenced in part by their genes. And so gene and environment may be not independent. There can be gene-environment interaction, obviously, and that might reduce the apparent heritability. More recently, people have recognized that there may be epigenetic effects such that monozygotic twins are actually more similar in their epigenomes than dizygotic twins, and that may create a phenotypic similarity that, again, is not entirely due to genetic DNA sequence variation. So just to give you an idea of the different numbers that we use here in case you're interested in how some of these things are derived, basically the overall phenotypic variance, that's this V, is going to be related to heritability, the genetic variance, shared environment, and individual specific environment, or actually this also includes measurement error. And so what you're looking at is how similar, and then you can express that as a correlation, are monozygotic twins compared to dizygotic twins. And just for your interest, the heritability is often calculated as twice that difference. So let's say that the, and you can use concordance rates to estimate these correlations as well. So let's say it turned out that the concordance rate of monozygotic twins for schizophrenia, let's say, is about 50%, and for dizygotic twins is maybe 10%. So what would the heritability be? Be 50 minus 10 is 40 times 2 is 80. And that actually is about the heritability of schizophrenia, and those numbers are about right. So heritability can range from 0 to 100%. And it's interesting to think about or to look at the differences of heritabilities in a variety of traits. So let's take something like, well, schizophrenia, I told you. What do you think is the heritability of autism? I think it's higher than 50% or less than 50%? Higher? Okay. And what about ADHD? Does people know what ADHD is? Attention Deficit Hyperactivity Disorder? I think that's higher than 50% or lower than 50%? Lower. So here's the estimated heritabilities of a whole bunch of psychiatric disorders. And what you see is that autism is really pretty high. These estimates range from maybe 80 to 90%. It turns out that ADHD, this is surprising to many people, actually has a pretty high heritability. And in fact, it's one of the most stable estimates. That is, almost all twin studies come to that same range. So it's actually pretty highly heritable. Anxiety disorders and so on. And I've put breast cancer here and even gene expression as a comparison because it highlights an important point. How many genes do we know about for certain that influence ADHD? Yeah, no reason you should know this, but the answer arguably is none. There have been a number of genes that have been repeatedly implicated, and there are some that have survived meta-analysis. How many genes, do we know of any genes that have been shown to influence breast cancer? Yeah, a long time ago. I mean, those were discovered fairly early on. And how likely is it that genes or genetic variation influences gene expression? Pretty likely. Okay, so why is it that something with such a high heritability genes aren't fallen out of the sky, and yet something with a much lower heritability, we long ago identified some pretty major genes? Anybody want to speculate on the answer to that? Complexity of the syndrome. Yeah, essentially that's right. So remember what this is, what the heritability is. It's the proportion of variation in the population attributable to genetic variation. So you could have genes like BRCA1, BRCA2, which have very highly penetrant mutations, and yet on their own don't account for most cases of whether somebody gets breast cancer or not in the population. And so they will contribute relatively little to the overall heritability. On the other hand, you could have a syndrome where there are many thousands of genes that each contribute a small amount and that add up to a pretty significant number. So the heritability is a little bit of a tricky concept. You might have intuitively thought that the more heritable, the better. Take something like height and weight, and you're probably all familiar with recent studies of this. But this, by the way, is the different movie stars who have played James Bond over the years, starting with Sean Connery, George Lazenby, Roger Moore, Timothy Dalton, Mel Gibson, and their relative heights. Has no connection to anything I'm really talking about, but I thought it was an interesting pearl. And weight and BMI. The point here is that height and weight are really highly heritable. So should be relatively easy to find genes for those disorders if heritability is a good index of how genetically influenced something is, although as we just talked about, it's not, because the complexity of the disorder can mean that you can get to a heritability that's high with lots of tiny effects. And this was pretty dramatically influenced by the recent studies that have come out of here and elsewhere, Joel Hirschhorn and colleagues, and the GIANT Consortium, looking at variants that influence height and BMI or weight. So just recently, pretty large study, 183,000 people, identifying 180 loci, which is pretty good, explaining 10% of the variance. And actually, this chart down here that you can't really see all that well is saying, using the information that we have, that there might well be, if we wanted to explain something like 15% of the variance, we could do that. There'd probably be, based on the data we have so far, about 700 variants, and we'd need about 500,000 people to do it. BMI, in some ways, even more daunting, 250,000 people explaining about 2.5% of the variance. So what that means is that even relatively simple phenotypes, in theory, that can be easily measured, very precisely measured, we've known how to measure height and weight for a number of years now, doesn't mean that it's going to be easy, and these are highly heritable phenotypes. And in some ways, if you think about it, maybe that's not so surprising, because the simplicity, the precision of the measurement and the heritability don't tell you all that much by themselves about the biology. And so what you're seeing here is a number of biological pathways and processes that contribute to these very simple, precisely measured phenotypes, right? Practically, I mean, if you think about it, height and weight are essentially the integration of practically everything that's going on in your body in development and throughout life. Your height actually does change not only to a maximum, but then starts to decline. And your weight obviously can fluctuate. So you're pretty much aggregating effects over potentially an enormous number of complex processes, each of which may have its own genetic architecture. So a few things about the heritability. First of all, just the concept that it is a population-based variance component. It is not a measure of how influential genes are in this patient or individual's phenotype. It doesn't mean if you have a heritability of 60%, that 60% of individual, that 60% of somebody's depression, let's say, is due to genes and 40% is due to environment. Obviously, it doesn't work that way. What it means is that since it's greater than 0, there's some kind of genetic variation going on, and 60% of the individual differences are attributable to individual differences in genetic variation. And importantly, it doesn't say much about genetic architecture, as we just saw and we'll return to in a moment. And it also depends on, because it's a ratio, the degree of genetic and environmental variation in the population. This is what I think one of the most striking sort of illustrations of this was a study that came out several years ago looking at the heritability of IQ by socioeconomic status. So here what you're looking at is A, we said that's the additive genetic component, here's the shared environment component, here's the individual specific environment component, and here's the proportion of the variance explained, and here's the socioeconomic status measure, let's say. So this is higher, this is lower. And what do you see? You see that as socioeconomic status goes up, the apparent heritability of IQ goes up, and the environmental effects go down, and vice versa, obviously. So it seems to be moderated by the environment. What would the interpretation of that be? How could something like IQ be influenced by something like socioeconomic status?
People of higher socioeconomic status have access to better information.
And so how would that explain why the heritability would go up? So you're saying that people with higher SES would have better environmental access, let's say, to things that might be. And how would that account for this curve?
I think it's just that at low socioeconomic status, the environmental factors just swamp any genetic.
Yeah, I think that's probably right. That at low socioeconomic status, differences in environmental resources, for example, could have a huge effect that swamps small effects of genetic variation in the population, and that maybe once you get to a certain point of environmental enrichment or access to resources, it doesn't matter. You kind of get a ceiling effect, and that's when you see these genetic variations uncovered. So, that's just an example of how you have to be careful in interpreting the heritability. First of all, it depends on the population you've studied, and it depends on the mix of environmental resources or features of that environment. And then just to drive home the point again that an overall heritability could decompose into very different kinds of genetic architecture. So here's the most simple kind of case. Let's imagine we have a heritability total of 75%. You could get that with 3 loci, each contributing 25%, or 30 loci, each contributing 2.5%, or anything in between. And actually, Neil Risch, a little while ago, sort of gave another example. If you got to 12% locus-specific heritability, you've got a variant that contributes 12% of the variance, you can get that from a Mendelian dominant that's rare but has a huge effect, or from a common variant and a, sorry, that was the rare disease, or a common disease, common variant that has a much smaller effect. So it's really not a reliable guide about the genetic architecture, which itself obviously is going to have major implications for how you do your study, right? If you think that it is lots of common variation of small effect, you're going to do one kind of study. If you think it's mostly rare, more Mendelian kinds of effects, you might do another. Okay, so let's move on to this issue of phenotype measurement. Let's say you've selected a phenotype, you think it's heritable, you now have to decide how you're going to measure it. And there are a number of issues that you could think about here because obviously, I mean, there's some sort of mom and apple pie issues. If you are imprecise in your measurement and you kind of wing it and you don't really have a good measure, then that's going to reduce your power. That's sort of pretty straightforward. But modeling the phenotype can have a big effect. So let's say you have, this is sort of what's considered the classic liability threshold model. The idea that you've got a trait that might even be normally distributed. And we in medicine, let's say, or in research, often draw a line, a threshold that says, okay, this is a disease and this is not a disease. And you could imagine that with blood pressure, let's say. People call hypertension, classically, 140/90 or above. And so the problem, as you can see, when you're making this kind of case control split, you could do a case control study of hypertension. All these people are your cases and all these people are your controls. But notice, of course, at the boundary, you've got these two people who are kind of indistinguishable in their phenotype, if you really think about it, and yet you're throwing them into two totally different groups. And often you will get more power under many circumstances by actually using the quantitative trait distribution, although under some circumstances you can enhance your power by looking more at the extremes. If you have enough people, getting the extremes can be more powerful. And actually, Naomi Ray and Peter Vischer and colleagues just sort of published some simulations showing this and showed that if you had this kind of model of underlying disease, where you have a genetic liability and a threshold, it turns out that the case control comparison is actually more powerful as long as your cases make up a substantial proportion of your overall sample, 20% or more, and the disease is not really, really common, so less than 10% prevalence. And what you're looking at here is the ratio sample size between a case control and a quantitative trait measure of a phenotype, assuming it's the same genetic architecture. So if this is less than 1, it means the case control is more powerful. And what you see is that the quantitative trait only gets more powerful when the proportion of cases is really pretty low, like 10% in your overall sample, which you probably wouldn't do. And your disease frequency, the higher the disease frequency, the less advantage of the case control. So the notion is that you're, in essence, enriching your sample for cases and therefore improving compared to if you just pulled people from a general population, which often we don't do anyway. But the sort of issues to watch out for are there are lots of ways you could misclassify people. And each of them can cause problems. And you're probably familiar with some of these, but it boils down to whether you're looking at the right phenotype or genotype. So you can have phenocopies, that is people who appear to have your phenotype of interest, but have a different, maybe largely environmental cause of that phenotype. We think of that sometimes in depression, where maybe there are some people who become depressed largely for environmental major loss kinds of reasons, and other people who may have a stronger genetic liability. Incomplete penetrance, of course, means that you might have the genetic liability and not express the phenotype. Variable expressivity, meaning that you might have the genetic liability but have a range of phenotypic expression. And you, when you are making your study design, have to decide, does this person have the phenotype of interest, or do they have a variant of it, or are they really something else? And genetic heterogeneity, of course, which means that you can get the same phenotype, but there can be a massive range of genetic influences that contribute to that. And that's going to create problems in your study design. So let's take an example of Alzheimer's disease. And let's say this is the universe. This is the state of nature. And there are some people who don't have it and some people who do have it. But when you do your study, you don't know the state of nature. You know what you have ascertained. And so let's say you have made a clinical diagnosis, because you can't do an autopsy on living people, and you have ascertained a bunch of cases and a bunch of controls, and you can see that you're going to get a little bit of it wrong. So you're going to have false positives. That is, people you think have Alzheimer's disease, but in fact they have something like multi-infarct dementia, something that looks very similar. And you can have false negatives. That is, people who you think are controls, but maybe, you know, if you'd studied them 6 months from now, you'd realize they're actually a case. So what is the effect of that? Well, here's an example of how misclassification can have massive effects. So this is sort of a simulation, but what you're looking at here is for a couple of different allele frequencies, if you were looking for an alpha of 0.05, which I use just for illustration, you're almost never looking for an alpha that is a significance threshold of 0.05, but let's say you are. You've got a disease that has a prevalence of 1%, and you want 80% power to pick up this effect. If you were perfectly in tune with the universe and always called the true cases cases and the true controls controls, you can do pretty well with a relatively small sample. If you start making diagnostic errors, you quickly can lose control of the sample size that you need and get nothing. And it may be that some of the trouble that people have in replicating findings with association to complex traits has to do with the fact that, you know, in theory it'd be great if everybody measured the same trait the same way with the same skill and the same precision, but that doesn't happen. And so it may be that variability in these measurements are actually contributing to this heterogeneity in what we see. Here are a couple of rules of thumb just for your interest in case you're planning a study and want to think about these kinds of things. The diagnostic error problem is greatest when you're kind of in the range of you have just enough power to find the effect. If you have a massively overpowered study, which trust me, you will never have, it doesn't matter so much. You have some room to go. If you have a really massively underpowered study, this is not your major problem. And so it plays a little bit less of a proportional role. About a 10% decrease in overall phenotypic accuracy means at least a 20% increase in sample size to keep the power that you need, which for those of you who are actually involved in mounting some of these studies and designing them and budgeting them and so on is not a trivial thing, depending on how costly it is to phenotype. If the disease is uncommon, false positives are actually worse than false negatives. That is, if you call somebody who's really a control a case, that tends to be worse than calling a case a control or including cases in your control sample if the disease is rare. And just to sort of illustrate that a little bit further, there are a number of things that you can do if you want to select controls. So we just talked about selecting cases for your study. But it matters a lot how you select controls, both for your budget and for your power. Okay, so let's say that you have a few different opportunities. One is you're just going to scoop up people in the population and take population-based controls. You're not going to screen them or whatever. That might be the easiest thing to do. You don't even have to interview people or run blood tests or do anything. Or you could screen people just to make sure they don't have the disease that you're interested in. Let's say it's diabetes. Or you could take hypernormal controls, people who have the most extreme end of the distribution, who not only don't have your disease of interest, but they are the lowest in the traits that predispose to that disease of interest. So it turns out that which of these strategies you can use depends a lot on the frequency of your trait. And the different bars here have to do with a trait that, a variant that explains either 0.25% of the overall phenotypic trait, that's blue, and the red is something that explains maybe 1% of the variance. And then there's a difference in whether it's light or dark based on whether it's a common or rare allele. You don't have to worry about that too much. Here's the bottom line. When the phenotype is uncommon, these two strategies do about the same. So if you just ignored and didn't screen your controls for a disease that has a prevalence of, say, 0.5% in the population, you're going to do okay, because by far most of your controls will be controls, and that will not throw you too much. If the phenotype is common, say about 20%, then the more you screen, the better, the more powerful you get. This is, for example, the sample size you need. So it's much lower for the B than for the A. And it's always best to do the C in terms of power, but that may not really be a feasible option. So just something about case selection. And lastly, in terms of measurement, error, and so on, in gene-environment association studies, basically the same principles apply, except now you have to worry about the environmental assessment as well. So what you're seeing here is what's your sample size requirement, depending on how good your measure is correlated with the true exposure, that is the environment, or the true outcome. So let's say you're doing a study of the genetics of chronic obstructive pulmonary disease, and you ask people, do you have COPD, versus maybe something better, like you actually measure their lung function. So that something better would have a better correlation with true disease. And let's say you're asking about the risk of smoking and how it moderates genetic effects, whether you, say, ask people, have you ever smoked, versus you measure cotinine levels in their blood and you do very detailed measurement. Bottom line is, as you might guess, you can have major power loss by either misspecifying the exposure, the environmental exposure, or the outcome. And you can see at the extremes here, we're talking about 100-fold differences or so. Okay, so one approach to phenotyping is you want to get as many people as possible. And for something like height and weight, that's obviously pretty feasible. But what if you have something that's not quite that straightforward? There are a couple of strategies that people use to deal with the complexity that we were just talking about. One is make it up in volume. And that's the strategy of maximize the sample size, to pick up these significant effects, because of course your power is a function of effect size and sample size. And the other is say, well, what if we got smarter about the phenotype and actually identified a phenotype that had a stronger genetic effect or influence? Could we actually do better with fewer, with a smaller sample? And this is where we get into this notion of subtyping and intermediate phenotypes. And the goal here is to reduce genetic heterogeneity or phenotypic heterogeneity to get something that will have a stronger effect size. And this has worked, obviously, in things like breast cancer or Alzheimer's disease, where you take early onset, these are really unique cases, subtypes of the disorder, that showed major gene effects. So it was by subtyping that those early successes occurred. The problem is you can do subtyping till your heart's content, right? You can create, I'm going to look at people who have diabetes that they got at age 11 when they, whatever. I mean, you can make up a whole enormous list of subtypes. And of course, there's the familiar notion that you're going to maximize your likelihood of finding false positives unless you're careful about correcting for them. And so there are strategies that you can use, like really being pretty careful about what's a reasonable subtype, being explicit about the number of tests, doing some kind of data reduction to not test every possible subtype, and so on. And of course, the multiple testing problem gets magnified in the era of full genome analyses, both GWAS and sequencing. So this is the kind of question that if you're actually on a study section, you might be facing. Which $1 million study, $1 million is probably optimistic, maybe $10 million study, would you want to fund? Would you want to take a study that has, I say 2,000 here, but let's say it's 20,000. It's a really cheap study. It's got a huge sample size because all you're doing is asking people about stroke and asking, have you ever had a stroke? Which might have some ascertainment bias to it, obviously. Or you're taking a case control sample in the hospital and you're doing this intensive phenotyping, but your sample is much smaller. Which of those do you want to authorize our taxpayer dollars to fund? I don't know. There's no real right answer, barring some more information about these traits, but it's a dilemma. And those who would argue that you could do as well, if not better, with the intermediate phenotypes take the position that if you could define an effect that is closer to what the genetic loci are actually doing than some clinically diagnosed or self-report or some looser phenotyping, then maybe you can identify larger effect sizes. Maybe instead of asking people about asthma, you measure their serum IgE levels. And the notion is that the genetic complexity might be increasing as you go from things that are closer or more proximal to the actual gene effect compared to what we in say, the doctor's office diagnose. And so people have suggested all kinds of intermediate or sometimes called endophenotypes to approach this problem. And you could come up with lots of these things that could be proximal measures or intermediate measures. And not all of them are worth looking at. Just because something is not a disease doesn't make it a better phenotype than a disease. And these are some guidelines that people have suggested if you're going to try to maximize your phenotypic effect size and choose an intermediate phenotype. It should obviously be well measured. It should be associated with the illness in the population and co-segregate with the illness in families. It should be familial. It should be heritable. And ideally, the whole point is that it's got something of a simpler mode of inheritance. And that's rarely actually demonstrated. It also should have a biological plausibility and so on. I'll give you an example from psychiatry again, where arguably this has paid off. This is a gene, the serotonin transporter gene, that has been implicated in lots of psychiatric and even some non-psychiatric phenotypes. This is actually the gene that makes the protein that SSRI antidepressants, Prozac, Zoloft, Paxil actually work on. So it blocks a transporter that reuptakes serotonin. And here's a schematic of the gene. And what was found is that there are two variants in the promoter of the gene, a short variant and a long variant. The long variant has 44 extra base pairs of sequence, and that makes it a that makes it a more efficient or more active promoter from a transcriptional point of view. So you're making more serotonin transporter here and less here. And you can sort of think of it this way. So here's the transporter on the end of a neuron. Here's some serotonin. It's picking it back up. The SSRI antidepressant blocks it. Some people who carry the short allele, if they carried 2 copies, let's say, would have less transporter, more serotonin sitting around. And I'm not going to go into all the biology of that, but one notion is that that might have effects on how we process emotional information. It's been related to depression, anxiety, and so on, but the effects have been very inconsistent. And one thought was the effects are inconsistent because when we're looking at these very distal phenotypes, we're missing a lot of the effect. If we could get something closer to what this was actually doing. And one way that's been explored is to look at actual brain function. So it turns out that if I showed you, as I'm doing now, people with emotional expressions on their faces, that tends to activate circuitry in your brain that processes emotion, and particularly the amygdala and the insula. These are regions of the emotional brain. And that kind of activation has been shown pretty repeatedly to be an index of anxiety, depression, and so on. And so the question is, could we find an effect with even smaller sample sizes and more consistency using this kind of brain endophenotype or intermediate phenotype? And just to cut to the chase, a meta-analysis suggested that, in fact, of 15 studies, 10 showed that effect. That is greater activation with the short allele, the one that is considered a risk factor for disease. And then, in fact, it explained about 10 to 20% of the heritability or rather the trait variance. But what's amazing is that the sample sizes in these studies have been really small. So the argument is that we are able to pick up this effect because we're looking at the brain, the organ of interest in psychiatry, and not just asking people, how do you feel? There's still debate about this, but it's at least an interesting example. Another example that's going to be maybe even more useful for using these intermediate or endophenotypes is to actually take things we know from GWAS studies, sequencing studies, and so on, and say, let's look at the biological implications or the functional impact of these. And here's just an example of a gene that had previously been implicated in schizophrenia with a massive sample size. And with a much smaller sample size, this needs to be replicated, of course, that variant in that gene seems to affect connectivity, the connections between the cortex and the emotional brain, to put it oversimplified. And what we may be able to do is sort of what I call endophenotype mapping. That is, take variants we know and sort of map what is their functional implication. How do they actually act? That's probably in some ways going to be a more useful version of using these. But the argument is still there. That is, if we can make better phenotypes, we can actually have a pretty substantial impact on power. And here's just an example of a phenotype that has a stronger effect size. This is nothing too fancy. You need a lot fewer subjects to find an effect. That's the bottom line of that. Now, there's controversy about this, and actually some people would say there aren't a lot of examples where this has worked. Jonathan Flint's group and others have, for example, compared the effect sizes in mouse studies of behavioral traits, which we would say are the more complex, and sort of biological measures like height and weight and so on. And the effect sizes are the same, and they argue, well, that suggests that endophenotypes aren't going to be that useful. Although, as we saw with things like height and weight, those may not be the most simple phenotypes either. So just to summarize so far, and I think we've got almost no time, because we got a little bit of a late start, so I may not go into the last part about these cross-disorder phenotype things. But basically, phenotypic selection, you want to find something that you really believe and can convince yourself and others is familial, is heritable. Obviously, you need to measure it reliably. And then you need to make a decision, which is often a practical decision about what you're going to do with that. Are you going to do massive large samples to overwhelm the noise of the phenotype, or try to do better than that? And these are really practical implications that have, when you're doing these studies, have tremendous effects. I will, it looks like it's 9:30 now, so I think the MPG meeting is about to start. So maybe we will save this for another time. The bottom line of what I was going to show you in the next couple of slides is just that we are now beginning to be able to turn this around and ask the question, can we learn from what we know genetically to disentangle the phenotypic spectra of disease? So first we were asking, what's the best phenotype to find the genes? And now we're asking, can we use genetic information to disentangle this kind of morass of phenotypes, which, just to give you an example, in psychiatry, looks like this. This is our diagnostic manual. That book I showed you, if you tried to look at it from what the genes are telling you, it really looks like a more overlapping mess. And there are increasing studies, I'll just say, in autoimmune disease, in psychiatry increasingly, and in other fields, that are finding that it's not one disease, one gene, one disease, and in fact there is tremendous phenotypic overlap, pleiotropy, if you will, in the effects of genetic alleles. The reason that's interesting is not only does it tell us something about the genetic architecture of these traits and so on, but it has implications for understanding what do we mean when we define a disease. How do we decide that this thing that we're drawing a boundary around is a disease and it's different from this thing? And that's an interesting sort of philosophical question. It's more than philosophical in psychiatry because we actually change our disease definitions fairly rapidly. But even in other areas of medicine, it's a fascinating look into common pathways and shared biology that may cause us to restructure how we think about disease. It doesn't mean that we have to, because obviously things could share risk factors, but we still think they're different diseases. There are lots of other things that play into that. So why don't we leave it there for now and put a promissory note on talking about that further next time. I think